This chapter covers titration by measuring pH. The objective of this experiment is to create a titration graph and determine the dissociation constant of a weak acid, or Ca. The weak acid we'll be studying today is maleic acid. You'll see that the lab provides X molar solutions. You need to find the value of X, and then we'll determine the Ka of maleic acid. The experiment consists of two parts. First, we'll standardize the sodium hydroxide solution. Second, we'll titrate by measuring pH. The pH measurement part will also consist of two experiments. First, you'll perform a rough titration to find a rough endpoint. The second part will be creating the titration graph. For the first part, standardizing the sodium hydroxide solution, you will weigh KHP, potassium hydrogen phthalate. The first method is to weigh 1.02 grams of KHP. Using a 4 decimal place balance, weighing it only once. Next, dilute the substance with distilled water in a 100 ml measuring flask. Then, pipette 25 milliliters of the substance 3 times into an Erlenmeyer flask. And titrate with sodium hydroxide. Alternatively, students can weigh 0.25 to 0.30 grams of KHP 3 times into this weighing flask. Dilute with 75 milliliters of distilled water. And titrate with sodium hydroxide 3 times. For weighing, use a 4 decimal place balance. Press tare first. Place the weighing flask into the balance. Keep one side open and all sides of the balance closed to prevent air from entering. Then press tare to 4 decimal places. This 4 decimal place balance is used for preparing standard solutions. Then add the substance for now. We'll use 1 gram if you're diluting the substance. Be careful not to spill it. This is an approximate value. You should get around 1.018 or 1.02. Record the weight here. For calculating the precise concentration of a KHP standard solution, to prepare a standard solution by dissolving KHP. After weighing, pour the solution into the beaker. Some solution may remain in the weighing flask. Rinse with distilled water. Then, add a small amount of water until the solution is completely dissolved, and pour it into the volumetric flask. After dissolving KHP, pour it into the volumetric flask. The pouring technique involves tilting the flask once. Leaving some solution above the beaker to check for any remaining residue. Then, pour glucose solution into the volumetric flask. Use glucose solution to adjust the volume of the solution. When adjusting the volume, ensure your eye level is at the volume mark. On the 100 milliliter volumetric flask, observe the lower curve of the beaker. Once you have the standard solution, pipette The small size of the volumetric flask. The mouth of the volumetric flask is quite small, so we'll pour it into a beaker instead. For this experiment, we'll be titrating 3 times. So, we'll pipette the solution 3 times into an Erlenmeyer flask. When pipetting, keep the tip of the pipette at the bottom of the container. For titrating the KHP solution with sodium hydroxide. First, prepare the sodium hydroxide solution. Pour it into a 50 milliliter burette. Rinsing the burette with a small amount of the sodium hydroxide solution beforehand. Then, prepare the sodium hydroxide solution by clamping. The burette, while adding the sodium hydroxide solution to the burette. Check for air bubbles. At the tip, if there are bubbles, release the sodium hydroxide solution into the receiving container and let it drain until there are no more bubbles. Next, have the students add the sodium hydroxide solution until the initial volume is 0.00 and record the initial volume. For titrating KHP with sodium hydroxide. First, have the students add the indicator drops. The indicator used today is phenolphthalein. To titrate KHP with sodium hydroxide, add approximately 2 to 3 drops. Then, shake to mix. Before titrating, slowly add the sodium hydroxide drop by drop. Shaking the flask containing KHP as you titrate. When the endpoint is reached, which is a pale pink solution, record the volume at the endpoint. Then, repeat the titration 3 more times, adding the remaining 2 drops. Therefore, you will have a total of 3 endpoint volumes. After that, find the average and calculate the concentration of sodium hydroxide. The second part is titration by measuring pH. First, before titrating by measuring pH, determine a rough endpoint by pipetting 20 milliliters of maleic acid solution into an Erlenmeyer flask. Then, add 2 types of indicators. The first indicator is methyl red, and the second is add 2 to 3 drops of phenolphthalein. Since maleic acid is a diprotic acid with 2 protons, we will have 2 endpoints. So we need 2 indicators. The first indicator added is methyl red, which will first change color to orange because it initially turns red when added to maleic acid. Then, we titrate from red to orange. Once the orange color is achieved, the remaining second proton will change color in the phenolphthalein range. Actually, phenolphthalein is colorless in an acidic environment. Since the initial solution already has the orange color from the methyl red, continue. Titrating with sodium hydroxide. This will initially produce a yellow color. But, as you continue titrating, you will regain the orange color, which is the second endpoint. Then continue titrating until you reach the second endpoint, which is the original orange color. After that, you will regain the orange solution. This is the second endpoint. Students should record the volume at both endpoints. If students continue titrating, they will exceed the endpoint and will obtain a red solution. This indicates that the solution has exceeded the endpoint. After students have roughly titrated to find the endpoints, the next step is to set up the equipment like this. A burette containing sodium hydroxide solution. A beaker containing 20 milliliters of maleic acid solution, prepared using a pipette. 50 milliliters of distilled water, prepared using a measuring cylinder. A magnetic bar placed on a magnetic stirrer. A pH meter. This is the pH probe for measuring pH. And a ledger. One student will be responsible for recording the experimental results. And another student will plot the pH graph from the data. After students have finished setting up the equipment, before using the pH probe to measure the pH of the maleic acid solution, students must calibrate the pH meter first, using buffer solutions. Ideally, 3 pH buffer solutions will be needed. pH 4, pH 7, and pH 9. However, today we will only demonstrate using pH 7. First, immerse the pH probe in the buffer solution, then on the pH meter screen. Select calibration. Next, select 1-point calibration, and then calibrate now. Since our pH probe is immersed in a pH 7 buffer solution, adjust the pH meter to read 7.00. Set the reading to 7.00, then click Keep. Finally, click OK. The pH meter should now read 7.00. Before use, rinse the meter thoroughly with distilled water, especially the center section. After completing the experiment, rinse the pH probe in the same manner before immersing it in the buffer once. Then, gently dry the pH probe with a tissue. Immerse the pH probe in the maleic acid solution. Next, turn on the magnetic stirrer so that the magnetic bar inside the beaker containing the maleic acid solution rotates. While the magnetic bar is rotating, observe carefully to prevent it from touching the pH probe. Students are to read the pH of the maleic acid solution before adding. the sodium hydroxide solution. Now wait for the pH meter to stabilize. After turning on the magnetic stirrer, read the initial pH value before adding the sodium hydroxide solution. Count to 5 seconds and record the reading. After recording the initial volume before adding the sodium hydroxide, proceed to add the sodium hydroxide. The volume to be added should be based on the endpoint volumes obtained in step 1, adjusting by plus or minus 5%. Near the endpoint, add 0.5 ml at a time. Add 1 ml at a time after each addition. Count to 5 seconds and record the reading. Should also record the reading. The student performing the plot should immediately plot the graph. In this manner, until 40 milliliters of sodium hydroxide is added. The student will obtain a graph which is called a titration curve. This titration curve will be used to find the equivalence points. Since maleic acid is a diprotic acid, students will obtain 2 equivalence points. The volume at these equivalence points will be used to calculate the pKa of maleic acid. Therefore, 2 pKa values will be obtained. The equivalence pH at point 1 will be used to calculate pKa1. Equivalence pH at point 2 will be used to calculate pKa2. Furthermore, the pH at point 2 will provide the volume, which can then be used to determine the concentration of maleic acid. Remember that the concentration of maleic acid from the bottle is written as X molar. Students must find the value of X